All right. Yeah, thank you for having me here. I will not be talking about collective behaviors. We're going to have a bit of a change in topic now, but I wanted to talk a little bit about the kinds of problems my lab is interested in, which is how brains organize behavior on multiple time scales. We're especially interested in kind of survival behaviors that biology needs to get right for animals to survive and reproduce. So I'm going to tell you one story that's published and one unpublished story that I've never given a talk on before, and I put the slides together last night, so here's hoping it goes okay. So yeah, we're interested in species typical survival behaviors. Today it's going to be mice, mostly rodent species, work in a variety of organisms though. To survive and reproduce, animals have to express a variety of behaviors, and these behaviors can vary across species. They can vary across individuals within a species, and they can vary within the same individual over time based on your past experiences. Perturbations such as sickness or stress or hunger shift the expression of your behaviors to adapt to your needs. And as a theoretical neuroscience lab, we're interested in how you build brains that can do this so that you don't just have one fixed species typical survival program, but you have phenotypic plasticity. You can adapt the space of behaviors that you express in response to the environment around you. I won't really be talking about the I'll show some neural stuff today, more so than the previous talks, but we're interested in what mechanisms you can build into neural circuits to allow this kind of phenotypic plasticity. Things that brains can evolve, like changing where you're expressing neuropeptides and neuromodulators, how different brain areas are connected to each other, the physiology of neurons, circulating hormones. So these are all kind of molecular tools that brains have access to shift the dynamics of neural circuits and shift how they process information to alter expression of behavior. So for today, I'm going to focus mostly on the behavior side. And I want to introduce this framework of thinking of behavior as this kind of closed loop process of interacting with your environment. So if you take an animal that's out surviving in the world, it is carrying out motor actions to modify the state of the world, and it is receiving sensory cues from that world. And so the goal of the nervous system is to take the sensory information and translate it to motor outputs, and to do that in a way that maximizes survival. So we can draw this as the traditional sort of closed loop feedback controller, where the sensory state of the world gets sampled by some noisy sensor that forms an estimate of the world state. That world state feeds into a controller or a policy, depending on whether you're control theory or reinforcement learning. And that policy decides on the action to carry out, which then gets filtered by or combined with whatever motor noise your system has to act on the state of the environment. So the classical example of feedback control is regulating the temperature of a house. So the house is your system. It has a temperature. We sense the system with a thermostat that lives somewhere in the house and is reading out the temperature of the room. That noisy sensation is used to drive an effector, in this case, a heater or a fan that is turning on and off to try to adjust the state of the house as a whole. So pretty straightforward. This is the problem that brains have to solve, except in the case of brains, they don't just need to process the sensory information and produce the motor output. There's also this problem of deciding your set point. So brains have to decide what temperature do I want the room to be. And this isn't necessarily like a fixed homeostatic process where you always want to produce X amount of a behavior, but brains can tune their set points to adjust behavioral programs in response to their needs. So we are kind of the homunculus that controls the behavior of this thermostat system, because we set the dial on our wall to tell the house what temperature it should be. Brains, this controller is built into the whole system. And so both the thing that sets the target of your actions and the things that respond to that target and carry out actions are all kind of bundled within the same system. So if we go back to mice instead of thermostats, we kind of have two pieces that are happening within the brain. So this is all kind of exterior to the brain up here, and then inside the brain. Brains have to form representations of the perceived state of the environment and of themselves. So we have both what's going on in the world around us, and we're performing inference in a noisy environment. But then we also have interoceptive signals. So we have hunger and thirst. We have maybe mental maps of the way our environment works and the rules that it follows as we interact with it. So a big part of the brain is just this inference and modeling problem of understanding how the world works and what we need. And then brains also have to carry out action selection. And what I'd like to argue is that there's kind of different levels of thinking about this. There is the moment to moment action of what you're doing. So whether you're eating or foraging or fighting another member of your species. But then there's also these internal motivational states that brains set. So how much am I prioritizing responding to pain or interacting with conspecifics or hunting for food? Sometimes these are homeostatic processes, but other times they're not. There can be kind of like feedback loops, positive feedback and negative feedback, that make these internal motivational states dynamic as opposed to just fixed targets that you're moving towards. So I want to talk about two projects today. The first is going to be dealing with these internal motivational states and how they interact with each other when different survival needs come into conflict. And then we'll talk a little bit about the more fine timescale action selection.
And quick question.
Yes.
Maybe it doesn't make a difference, but where we've drawn it, the internal motivation state, in some sense, sends some vector to the action selection, but in principle, it could also influence the perceived
System state. Yeah, you could have like a predictive model of like.
No, I also meant the perceived. Anyway, maybe it doesn't matter in the final analysis, but the top arrow could also be influenced by your internal motivational state.
The top arrow.
The perceived system too, yeah.
Oh, like your representation of the world. Yeah, I guess I'm treating this as more like emotional states as opposed to interoceptive states.
Is it okay to be additive rather than some multiplicative?
Quite possibly, yeah. This was sort of just to evoke the idea of like a hierarchical controller where you have big picture goals and then you have how you're getting to those goals as kind of distinct computational problems that brains have to solve. So this is a published story I'm going to talk about in collaboration with the lab of Nick Betley, looking at interactions of survival needs in the brain and how they shape behavior. And this is in the context of pain. So when I started this project, I didn't think pain was all that interesting. I was like, it's another sensory system. But what's interesting about pain is it's almost more like hunger or tiredness than a pure sensory input. It's very sensitive to your mood and what you're doing, whether you're expecting a stimulus to be painful or not. There's a lot of affective aspects to how we perceive and process and respond to pain. And in clinical drug development, what people are really interested in is understanding non-acute pain. So like the way we study pain in the lab, it's usually like a pinprick or a hot plate. The pain that people are trying to take opioids or drugs for is not pinprick or hot plate pain. It's this sort of sourceless chronic pain that occurs. And we're interested in how that kind of chronic pain state emerges in the brain and how it can be modulated. So one way that people study this in the pain field is through assays that cause kind of longer term injury or tissue damage. And one of those is the formalin assay. So you take a mouse, you give it this formalin injection into the hind paw. I feel really bad describing this to like a mathematics audience. The neuroscientists are like, yeah, sure, okay, but pain research. So there's an acute pain response to the needle prick itself, and then there's kind of animals will lick their paw and respond to this sensory stimulus, and then they'll kind of stop and go back to baseline. But then around 15 minutes after this injection to an hour, there's this secondary pain response where there's like a lot of burning and tissue damage, and there's a secondary phase of behavioral reaction to the pain. So, sorry. If you look at a mouse during one of these assays and you just count how many times it licks its paw, you'll see that there's this initial period of high licking that drops down to baseline, and then there's a secondary inflammatory response that clears up by the end of the hour. And if you look at brain areas that receive ascending pain input from the spinal cord, they show activation that kind of mirrors that. There's this initial response. This is the parabrachial nucleus, which just sits on top of the dorsal horn of the spine and gets input from the spine. It gets this early response and then this later response that kind of mirrors the animal's behavior. So why are we looking at this? Because the Bentley lab several years ago, our collaborators found that if you repeat the same experiment in a mouse that's hungry, it loses that second phase of the pain response. It's pretty much abolished. You'll do this injection of formalin, they'll lick their paw for 5 minutes and then they'll stop. And then there's kind of nothing for the rest of the test. And if you look at the videos of these mice, they're pretty active. They're maybe even more active than the normal mice, and they're just not really responding to the secondary inflammatory source of pain. So you're seeing that in the gray trace is the licking behavior, and then the green trace here is the activation of the parabrachial nucleus again. So there's this initial response and then just kind of flat. Nothing's happening. And in 2018, Bentley Lab showed that this was arising due to a projection of AGRP neurons to parabrachial nucleus. So the agouti-related peptide neurons are in the arcuate nucleus of the hypothalamus. And when an animal is hungry, they start to produce a neuropeptide called neuropeptide Y. They have this hub and spoke interaction with the brain. So there's like a big condensation of these neurons in the arcuate nucleus, and then different subpopulations send out projections to different parts of the brain. There's a projection that reduces reproductive behavior and delays onset of puberty. And there's this projection to the parabrachial nucleus that seems like it's gating out this ascending pain signal. So these neuropeptide Y neurons release NPY onto parabrachial neurons that express the NPY receptor. And it's this release that seems to be necessary for the suppression of pain behavior. So in follow-up work, the Bentley lab looked at the system in more detail, and they found that it wasn't just hunger that had this effect. If you expose a mouse to predator odor, they also have an initial pain response to the formalin injection, but then they don't show pain coping behavior for the entire rest of the hour. TMT is a molecule that's found in fox urine. It's sort of innately aversive to mice. But these mice aren't just like freezing for an hour. They're still moving around and doing things, but they're not responding with pain coping behaviors. And the Bentley lab actually found that there's a separate part of the brain in the subfunicular organ that has a set of neurons that produce neuropeptide Y in response to TMT and also project to PBN. And so it seems like there's kind of this convergent mechanism that's settled on using NPY input to PBN to block out pain coping behaviors when something else more important is going on. So normally if you have an injury and there's this burning in your paw, you want to go back to your nest and lick your wounds and recuperate. But if you're starving to death, you're going to die if you do that. And so your brain has built in this contingency plan that basically shuts out the pain signal so that you can go out and forage for food. So my former postdoc, Amadeus Mays, and I asked from a computational perspective, how do we make sense of this? So we have this kind of feedback controller that's looking at your ascending pain signal from your spine and is deciding what to do about it. And we figured a maybe reasonable way to formulate this would be to say that the goal of the animal is to, it always has to make a choice about what it's doing. And anything that it does has an effort cost to it, that I'm responding to stimulus X at the cost of being able to do anything else. And when an animal decides to lick its wound, it is expending effort to reduce its pain. There's evaporative cooling, there's a mechanosensory blocking of pain signals. So you can choose to expend effort and reduce your pain percept, or you can choose not to lick and allow that pain to grow over time.
So what happens? It grows.
Because there's this ongoing sensory input coming from the periphery.
It doesn't reach so much stuff.
Can't really ask the mouse.
For people.
For people, it continues to be not fun. For a while, you have this
Grows and grows.
hard to quantify. It persists, at least. I don't know if it grows linearly. Continues to integrate. So every time you have a bout of licking, if you have these effort and pain signals, you can expend a little effort, exert a little effort and increase your effort expenditure to reduce your pain. And then when you stop the behavior, these two things will recover. So then if you come up with this formulation, you can say what optimal behavior is, which is to minimize your pain with minimal effort. So only like if you're in a regime where it's going to help reduce your pain, and when pain has reached a certain amount, that lick is contributing to that reduction. So we write that as this negative reward where the reward you're maximizing is the sum of pain and effort, or this cost that you're minimizing. So if we formulate it as this feedback process, then we can ask from a computational perspective, say we have a controller that works like this, and now we want to introduce a competing need state. We don't just have pain and the effort we spend to respond to it. We have other things like a drive to eat or to respond to a threat. Where should it enter into this system in order to change pain coping behavior and shift the animal's attention towards other things? So you could imagine that it gates the perceived pain intensity of the animal, that it just shuts out input to the system entirely so that the controller never sees anything to respond to. You could also predict that it changes the energetic cost of performing a behavior. You're starving, you have less energy reserves. So when you decide to respond to your pain, that costs you more than if you were relaxed and sated and everything was going well. So maybe what hunger is doing is it's changing the cost of actions that aren't related to foraging. Or maybe you have some behavioral control policy that doesn't just take in pain and respond to it, but it takes in some nonlinear or linear or nonlinear combination of your pain signals and your hunger state, and adds those two together and weighs them off against each other to make a decision. So could we add maybe gating of motor output, or add a term in our cost function that scales these things depending on scales pain and effort depending on hunger state? So we formulated this model. We could train a little neural network to reproduce the baseline pain coping behavior. So we make up an input to the model, an acute response and a long-term, or an acute input and a long-term input, and could get the model to produce licking bouts that kind of follow the two-phase response that we see in the mice. This is really just handcrafting an input so that the model behaves the way we want it to. This is now the model licking bout behavior, quantified the same way we did in the animals. But since it's a model, we can also see the internal state. We can see the effort over time and the pain over time. So remember, higher pain is pain, higher effort means you've expended more effort recently, and you're trying to minimize the sum of these two things. So the model learns a policy where there's sort of this threshold that in pain and effort space, where if your pain increases, you start to respond. That response, once that costs you effort and pushes you off of this decision boundary, and then you'll like recover back to it eventually. Yeah.
So there are some parameters in your read.
There's a lot of parameters, yeah.
And in particular, there's the relative magnitude of effort compared to reduction in pain.
Yes.
And the discounting factor. How sensitive, for example, at least to those three, are your...
Not terribly sensitive for the magnitudes. Like basically, no matter how you set them up, your model is going to kind of live on this decision boundary and just bounce around it. So your threshold for like how high your pain gets before you start responding to it changes, or like the exact bout structure, but like qualitatively, it was pretty well behaved for a decent range of parameters.
An effort over here is defined as motor effort, trying to get away, or...
An abstract cost that the model pays to produce a lick bout. And so we just like, it increases when the lick bout is triggered, and then that cost decays away exponentially over time after that. That makes sense?
What does licking do?
Yeah, it produces evaporative cooling, and that lowering of temperature alleviates the pain. It cleans the wound, and there's this interaction between mechanosensory fibers and pain fibers, where if you activate mechanosensory fibers, it'll actually gate out the ascending pain input at the level of the spinal cord. And so just by like rubbing your bruise or something, you can block out some of the pain signal from propagating up into the brain.
The reason I ask is, are there alternative approaches where you had
Yes.
cool environment and the animal could get there, and how would it essentially have a trade-off associated with different modalities?
Yeah, so I'm not going to talk about it today, but one of the first thing that we did is we had we had annotations for both this licking behavior and for paw guarding, where they just like take the right off their paw and stand there. And we wanted to model how they switch between the two. It was tricky, like getting multiple thresholds for different behaviors and getting it to match the way the animals like picked different behaviors for different stages of the assay. Spent a lot of time on it, ended up just putting it off for now, but it's something we're really interested in. If you have multiple actions you can perform that have different costs and different benefits, how do you select among them? And can we infer those costs and benefits from the animal's behavior? If you would like to work on that or know somebody who does, it still bothers me that we couldn't get that to work out. So just with pain and yeah,
Is one who tried to model pain, and if so, is that even possible given the behavioral data?
to model pain,
Can I
yeah.
follow up on that?
So for us, it is just a variable in our model that is going into this behavioral policy. So we can model it. We don't know the ground truth pain that the animal is experiencing. We just see its behavior. This is actually a huge problem for the pain field, that you can only know what an animal is feeling if you look at its behavior and it like reports it to you. I actually reviewed a paper on pain once where they claimed that they presented like a looming disc to a mouse, which induces this really strong freezing response. And they claimed that it had a analgesic effect because the mouse stopped licking its paw. So yeah, nobody knows what these mice are feeling. In the paper, which I'm not going to get into, we actually look at a subset of neurons in parabrachial nucleus that are super correlated with this pain signal. And so we could maybe try to use that as an objective measure of pain without having to read out something from animal behavior. But there's a lot of work in computer vision and machine learning trying to get more granular, like looking at facial expressions. I don't know if Bob Dotto will talk about any of the Mosaic stuff with like, he's worked a lot with Ishmael Abdus-Sabur and just like changes in statistics of behavior that could be indicative of a pain state. But it's yeah, you never get to see the internal state. You just have to guess. And pain is also, there's like this whole literature on animals hiding their pain, like racehorses with broken legs that hide their pain. And so it is an ongoing challenge in the field, which is why developing new pain medications is difficult. Okay, so this is our model formulation. And then we introduce our different hypotheses. So for that, I'll give you some equations. This is kind of gross. We wanted to capture the fact that competing needs could suppress the persistent but not the acute phase of pain. And so we built in this kind of hacky nonlinearity in the pain state that is probably unnecessary. But the basics of the model are that when you produce a lick bout, your effort increases by some amount and then decays over time with some time constant tau. Your pain is this integrated signal passed through a nonlinearity so that basically your pain saturates at some point. You can't just become infinitely more in pain forever. Within that integrated term, we have a decay at some rate, an ascending pain input that we just kind of pick up, and an effect of licking on pain. And this guy kind of like here is the combination of the linear and the nonlinear parts of pain. This is all kind of contributing to allowing the model to saturate, but you don't really need to think too much about that. So then given this model formulation, we can introduce these different modifying terms with this hunger element H that can be either 0 when the mouse is sated or 1 when it's hungry, and can change the dynamics of these variables and the way that the model behaves. So I'll start with what we thought it was going into this project, which is that the animal is hungry, so there's an increased energy cost to performing any behavior, including licking its paw. You can do this a couple ways. What we did here is just increase the time constant so that when you expend effort, it takes a longer time for it to recover back to baseline. We can do this in the model and we can say, how does the licking behavior of the model change? Turns out this wasn't a really effective way to change model behavior just because it was such a long-lasting input. This is really just like an ongoing input to the system over the course of an hour. So if you're changing these time constants on the order of seconds, you're not going to get dramatic shifts in how the model behaves. You're basically going to wait like a little bit longer to start behaving, and then you're going to just initiate the behavior. So no real dramatic shift. Sorry, the gray line is sated and green line or colored line here is under the hunger condition. We tried a couple ways of messing with the behavioral policy, just like changing the weighting between, sorry, this is like a proxy. This is the pain term that gets modified by hunger, or the effort term. So pain term contribution to hunger to the policy goes up or down. That was also not really effective at changing behavior, again, just because of the long time scales. Once you hit that threshold between like the licking and non-linking regimes of the behavioral policy, you just kind of bounce along it, and the dynamics it takes to first reach that threshold just don't matter all that much. You just kind of hover along it, and your movement dynamics along that boundary don't really matter. But if you gate out the ascending pain so it just doesn't reach the system in the first place, that's a very effective way of blocking behavior, which is kind of obvious in retrospect, that if you just don't perceive something in the first place, you're not going to respond to it. So from our modeling perspective, it seems like just blocking out a sensory input is the most effective way to change the output of a feedback controller, as opposed to trying to mess with the controller itself. This is also interesting because it might be something we see in other systems. For example, I think changes in olfactory receptor or pheromone receptor expression have been linked to changes in social behavior in rodents by folks like Catherine Duloc. So it could be a general principle that if you want to build flexible brains, the best way to do it is to just build in that state-dependent modulation as early in the circuit as possible. Yeah.
So I think you're only like invoking the gating for the second phase, or like what's?
The gating is there throughout.
Okay.
The nonlinearity that we hacked in there is what prevents it from really reducing the response in the first phase.
I see.
In the biological system, we think that there are different nerve fibers conveying the acute early phase and the sustained long-term phase. Different neural populations are activated, and a subset of them, just the long-term phase, are responsive to it. When we modeled this, we wanted to see if it was possible to do it with just a 1D input signal, which works so long as you have this like saturating nonlinearity and a very strong initial input. So basically the input in the first phase is so strong it hits the saturating part of our pain nonlinearity F. And then when you scale it down by this gating term, it's still high enough that it's able to drive the system. So you're just kind of moving within the saturated regime of a nonlinearity. The other thing that we can do with these models, we've said that we can find one that matches behavior and two that don't, is we can look at the internal state of the system. So the model's pain and effort state, and see a little bit more what's happening with these modifications. So yeah, gray is baseline, colored is hunger. You can see that for our behavioral policy model, if we increase this hunger term that reduces the weighting of pain, the model is in a higher pain state in the hungry case than in the sated case. It sort of waits until it's in more pain before it starts responding, and then it responds like it did before. Effort expended is about the same, a little bit lower. If we increase the cost of performing a behavior, we see something similar. You're waiting longer to act, and so you're experiencing more pain before you act. And so the model pain state is higher, and because the effort cost is higher, the effort state is also higher. And again, this reduction in perceived pain intensity makes the opposite prediction, that by gating out the ascending input, you're just not having a pain state in the first place, and because you're not responding to the pain, the effort state is also reduced. So this really just goes to show that different manipulations of different parts of a controller can give you very different predictions about what the internal state of the animal is. That the input gating hypothesis is saying that the animal is actually in less pain that we can't observe directly, but the other two are really predicting that there's more pain and it's just being ignored in favor of other priorities. So they make very different predictions about internal state. And from the imaging in parabrachial nucleus, we saw this kind of loss of neural activity during the secondary phase. So if we can interpret that as a pain state, then that's also consistent with this gating model. Again, you never really know for sure what internal states are, but it seems like this manipulating of the perceived state is really an effective way of controlling the system. So I have 15 minutes, I think. Awesome. So now I want to talk about the action selection part, and I've never presented this before, so we'll see how this goes. So we've talked about internal motivational states. I talked about pain today. I've looked at aggression in the past. They're these slow evolving things that aren't really linked to the moment-to-moment action or not action. And oops, that didn't work. Oh, okay, that was supposed to be an adorable GIF, but it's not. There we go. So animals can only do one thing at a time, and they have to decide what they're paying attention to. I didn't have cool videos, so I have this dog. We have to set our priorities of what we attend to in the world around us. And we get better at that with some experience, maybe. But when we're dealing with these competing survival needs, we have to integrate them all in and choose what we're paying attention to. And we have to switch between those things that we're attending to and the actions we're performing fairly quickly. So how does the brain do this? And what does this action selection thing look like? And how does it compare to the motivational states? So this was a collaboration with Li Ye at Scripps, who studies feeding and metabolism and cell energetics, and his grad student Tianbo Qi. So they were interested in brain areas involved in control of feeding. And the classical one is the neurons we just talked about that release NPY, these AGRP neurons in the arcuate nucleus. They're interesting. They look kind of like a hunger signal, but not quite like a hunger signal. So if you image the activation of these neurons in a hungry animal and you present them with food, their activity actually drops when food becomes available before the animal actually starts eating, and then stays low as the animal continues to eat if it is allowed to eat. So their activity looks like this macroscopic motivation to feed, but it doesn't really have anything to do with the fine timing of like, when am I eating, when am I not eating? So you have these bouts of eating, these interbout intervals. This is a mouse that was fasted overnight, so it's hungry and it's given access to food. And despite the fact that it's hungry, it doesn't just like eat constantly. It's until it's full. It'll eat for a little while and then it'll stop and wander around and it'll go back to eating. And the AGRP neurons don't really seem to have anything to do with this toggling behavior. Their activity is high before eating and it's low after eating. But if you look at their activity during eating versus during the interbout intervals, there's not really a significant difference. So Tianbo was looking for other brain areas that are modulated by feeding, and he used a tool from the lab to detect inhibited neurons, which I guess that's a bad animation spoiler. So the experimental setup is that you take an animal that's fasted overnight and then refed. It's given access to food. We control that animal. We compare that animal to a control mouse that's just given ad lib access to food. And we look at 3 different time points. We look at the mouse that had access to food, the control, a mouse at the end of fasting, and a mouse after this refeeding period. So fasting and then given access to food. And they did a screen for various reasons of neural activation in different parts of the hippocampus. There was some old literature on patient HM that he had trouble gauging how much he'd been eating. So they thought maybe hippocampus is involved in regulation of feeding. And they developed a method for labeling neurons that were activated or inhibited by feeding. So if you're a neuroscientist, you're familiar with CFOS. When neurons become active, these immediate early genes start being transcribed, and you can stain for CFOS and identify in a brain slice afterwards which neurons were active within the preceding hour or so. The Yale lab identified pyruvate dehydrogenase phosphorylation as an indicator of the opposite. A neuron that is active and then goes silent for a period of time can be labeled by staining for this phosphorylation of this dehydrogenase. So they looked in different regions of the hippocampus for this fluorescence of pyruvate dehydrogenase in the control and then the fasted and the refed. And they found that the dorsal subiculum of hippocampus had these neurons that were silenced after refeeding. So opposite direction of CFOS, the higher the bar here is, the less activity. So here's baseline in a fed mouse. When the mouse is starving, activity goes up, and then after it refeeds, the activity goes down. So it's a brain area that's silenced by feeding. They looked into these neurons with fiber photometry, imaging either in dorsal subiculum or in the dorsal subiculum neurons that projected to the mammillary bodies. They did a couple of other areas too, but I'm focusing on this projection for now. And what they found was this really weird kind of bimodal distribution of activity. So when the animal is eating, activity is low. This is just Z-scored bulk fluorescence of these neurons. When the animal stops eating, activity goes back up, and then it drops back down again when they eat. So it's this weird, like, bout locked activation of this projection, which is very different from what we saw in the AGRP neurons. So no difference in activity in the fasted state versus after 20 minutes of eating, but this big difference in activity during eating versus in between bouts of eating. So this was really wild. I'd never seen anything like this before, and it seemed kind of like something that was involved in this toggling between feeding and not feeding.
And
Yeah.
maybe it's reading tea leaves, but it looks like the activity starts going up in anticipation of stopping feeding.
Possibly. Oh, I have that on here. So if we just did, yeah, no worries. This was a surprise for me too. I just put these slides together. So if we trigger on the initiation of feeding or the termination of feeding, each of these gray lines is one mouse, and then the colored line is the average across mice. It's a pretty clean correlation with the behavior onset and with the behavior offset. This is GCamp, so it's not super good time resolution, but much tighter correlation than you would see in like archaeonucleus.
There's some non-causal filtering or something that's leading it, that's.
To the
what's the
anticipatory
cause?
rise,
Yeah, why is it non?
that it rises up before termination. First, human annotation of when you stop feeding is noisy. Second, if it is a motor command to stop eating, you would see it before the eating actually stops. I don't know why hippocampus would have a motor command to stop eating, but yeah, it's strange and unexpected. But it seems fairly consistent that you get this dropping off of activity. We looked at other explanations for the signal. Locomotion speed was not as good a prediction. We thought maybe, okay, you're active when you're running around and you're silent when you stop. But if we used speed as a regressor to try to account for this fiber photometry trace, it didn't do as good a job as just feeding versus not feeding. So the regression coefficient was lower, the variance explained was lower for speed versus feeding. They tried a bunch of other assays, which is kind of grossly shown here. So this is the fiber photometry trace I showed you before. This is the activity at the initiation of feeding. So like 0 is the average of the trace, and we're saying you're a little bit below your average activity right when you start to feed, or like when you're feeding. They tried other cases, so feeding when you're not hungry, feeding when you're cold, high-fat diet, liquid diet, and water all had the same kind of bout-locked response. If you gave the mouse a cotton pad to tear up and build a nest, so again, like hand-mouth coordinated behavior, you didn't get modulation. And if they were interacting with other stuff, like an object, a male or a female, you didn't really get modulation. So just specifically ingestion behaviors, regardless of context. So the other wild thing is that they took this population and they tried to manipulate it optogenetically and say, is this controlling how much you eat or some metabolic thing? They didn't get any clear differences in the amount of food that animals ate if they stimulated for the 20 minutes of the refeeding period, but they got weird changes in the bout structure of behavior. So if you silence this projection out of the subiculum, your feeding bouts were longer. You would eat for a longer period of time, and then the interbout intervals were not changed. So eat for a longer time and then go off and do something else and then eat for a longer time. So there was this significant shift right in the CDF of bout durations. And if you activated the projection, you got the opposite. There's a bit of a floor effect here because bouts can only get so short. But if you stimulate this projection, they eat for shorter periods of time before they stop. And there's no real change in the duration of pauses between feeding bouts. So not only does this projection track this toggling between eating and not eating, but you can activate this projection and change the animal's bout switching statistics, which is a weird thing to find in the dorsal subiculum, which previously has just been described in terms of place fields. And Korsakoff syndrome, which is a memory loss associated with a vitamin deficiency. And feeding. The other thing that we noticed was that there were sometimes these little transient events that happened during feeding bouts in the trace here. So Tianbo went in and just looked at the animal behavior and annotated for times when the mouse was eating and just kind of stopped and then went back to eating. And they coincided with these transient blips in the calcium trace. So when you have bout termination, your activity goes up and stays up. When you have these little interruption periods, you get this little couple seconds event that happens. And then the activity goes back down and the animal stops eating. We can look at the distribution of these interruption intervals. So all of these yellow periods here that are bookended by either the boundaries of the bout or these interruption events. And if we take intervals that led to an interruption versus intervals that led to a termination, they followed the same distribution. So they seemed like they were sampled from the same underlying process. You could think of it, and we thought of it as like a renewal process. You're eating and you get these little blips that come along. The distribution of those intervals was log normal. And each time you get a blip, you either go back to eating or you stop eating and go do something else. So that's kind of our mental model for this behavior is you have a mouse that's eating, something gives it these pulses that make it stop and look around. And then you're kind of bistable. When you get an interruption, you can either go back to feeding or you can switch out of the feeding bout and go to do some other behavior. Yeah, Kristen.
Is there any kind of reason it's supposed to pause while eating every now and then?
Attending to your environment, I think.
That's
Just
like a
like
digestion reason or something?
a digestion reason?
Yeah.
I don't know. Just enjoy the meal.
Okay.
I don't know. Haven't read deep into that. I guess chewing is noisy. Maybe you're just stopping to monitor your environment and make sure you're safe because you're a tiny prey species. And then you're like, oh, it's okay, and go back to eating. So we went out on a limb and just decided to model this as a bistable system, receiving perturbations from a renewal process. There's lots of bistable systems in math. We made it a Wilson-Cowan model because why not? So we have an excitatory and inhibitory population, recurrently coupled. This system has a phase portrait with two stable fixed points separated by this unstable fixed point. So you can be in a high activity state or a low activity state. If you're in the low activity state, you match that to feeding. And then this is to not feeding. And then you can feed this thing little kicks of input. And if you tune the distribution of those kicks, you can give, and you feed it with a little bit of noise, you can get these little simulated traces that look kind of close enough to the real thing. So you're kind of hanging out here and you're eating, and then these renewal events come in, kick you over here. And then if you make it past the separatrix, you will go into the stop eating state. Otherwise you'll decay back in. So having done that, we can say, okay, under this model, what is the optogenetic perturbation doing? Hypothesis one is that we're changing the frequency of the kicks. If you kick more often, then if it takes an average number of kicks to get past the separatrix, you'll get out of the feeding bout sooner. The other thing it could be doing is changing the threshold for transitioning out of the feeding state. So this is like a sideways energy well diagram of the feeding state and the non-feeding state. This is, I guess, the activity of the excited overpopulation on the y-axis, time, and these are kicks. So if you lower the depth of this attractor, then it takes a smaller kick to get you out of the feeding state. So the time statistics of the kick don't change, but the stickiness of the feeding state does. So we go in and we look at our optogenetic data, and we got a surprisingly clear result. The frequency of these interruptions and terminations was no different in the inhibition case or the excitation case, but the number of inputs you got before you ended a feeding bout shifted. So when you were inhibiting, it took a larger number of kicks before you stopped. And if you're exciting, it took fewer kicks to get you to stop. But the interval distribution between the kicks was no different between the two. There's a follow-up experiment in the paper, which I didn't throw in here because I'm still thinking about it, where instead of this optogenetic perturbation to the system state, which seems to be messing with the energy wells, we introduced a looming disk. That seems to increase the rate of the interruptions rather than the number of interruptions it takes. So if you have like more going on in your environment that you need to be monitoring, you pause and pay attention to it more often. Whereas if you're just manipulating the excitability of these cells, you're changing the depth of this attractor state for feeding. So this suggests that maybe multi-stable winner-take-all dynamics in subiculum could be providing this kind of stickiness to behavioral states. And it suggests mechanisms for modulating the stickiness of those states. So we think of hippocampus as a place that encodes a representation of our affordances and the cognitive map of our environment. It seems like that could be a useful thing for deciding when you toggle between things. If you have something in your head that's giving you a sense of like, what are the things I can interact with in the world? What do I have access to right now? That's kind of the information you would want to use to integrate with your drives and say, okay, now it's time to eat. Now it's time to run away from a predator. There's also some fun literature on hippocampus as predicting the consequences of actions and sort of like a model of state action pairs. So maybe it kind of makes sense that it would be involved in this kind of short timescale decision-making process. And it suggests that the way that you manage your bout statistics and action selection is by changing the stickiness of your attractor states associated with different behaviors and changing the rate of perturbations that could kick you into different attractor states. This is the case, seems consistent with what we see for feeding. The other reason I was really excited about this project is ventral subiculum is the single largest source of input to VMHVL, which is involved in aggressive behavior. And we never really looked into it when I worked on aggression. So subiculum sends projections to a lot of parts of the brain. And I just really like this. It's not very deeply studied, but I love this idea that it could be this kind of like job scheduler of the brain that's getting motivational state information and then toggling the animals between different actions. Having never worked on hippocampus before and knowing nothing about the field. So with that, I will thank the folks who did the work and thank you guys. The hunger and pain work was my former postdoc Amadeus Mays, and then the feeding work was with Tianbo Chi, who is recently graduated from the lab of Lié. And thank you guys for your time.